Hello friends, welcome to BMH Learning. In this video, we are gonna talk about isolation of lecithin from egg yolk. Lecithin is a type of phospholipid found in egg yolk. Requirements: acetone, petroleum ether, chloroform, ethanol, eggs, centrifuge tubes, measuring cylinder, conical flask, centrifuge machine. Procedure: first step is the isolation of egg yolk from the egg. So we are gonna use one egg. First of all, you have to break the egg and get the innermost stuff, that is egg yolk, into the beaker. Discard the other stuff containing the albumin. Egg yolk is actually in the form of a mixture made by constant stirring. Next thing we are gonna need is acetone. So measure around 25 ml acetone in a measuring cylinder. Add this much volume of acetone into the beaker containing egg yolk. As soon as you add the acetone into the beaker with egg yolk, the egg yolk start to precipitate. Mix it completely using a stirrer or a glass rod. Once the contents are mixed properly, keep the mixture aside for around 10 minutes. Next we are gonna centrifuge the egg yolk and acetone mixture. So the material is transferred to the centrifuge tubes. We are gonna centrifuge it at 4000 rpm for 5 minutes. When centrifuged, the two components, that is the egg yolk, precipitate and acetone, gets separated from each other. The upper layer is acetone and the lower one is, of course, the egg yolk precipitate. The supernatant is discarded. Now, once the precipitate is collected at the bottom of the centrifuge tube, remove the precipitate and keep it in a petri dish. So this is our crude egg yolk extract. Next we are gonna do is washing. The washing is done in order to solubilize different components of egg yolk. So for that, let's add the crude extract into a beaker containing sufficient volume of acetone. Mix the acetone with crude extract by constant stirring. Once the components are mixed, the suspension is subjected to filtration. So the contents are filtered out using a filter paper. Now keep one thing in mind. We have to repeat the washing step again and again until the filtered liquid which we are getting in the beaker becomes colorless. Apart from this, you can also notice the precipitate becomes white. Once the liquid turns colorless and the precipitate become white, this means the washing is complete. Next step is extraction. So we already know the precipitate which we are getting here is containing a mixture of components along with lecithin, which we wanna isolate. So in order to isolate lecithin from other lipid components, we need to extract it from the component mixture. So for that, we are gonna prepare 30 ml extraction solvent containing chloroform and ethanol in 2:1. So that means we have used 20 ml chloroform and 10 ml ethanol. Now pour this extraction solvent into the beaker containing white precipitate. Mix it properly and keep the beaker for 3 hours for extraction at room temperature. Once the extraction period is over, filter the content of the beaker using a filter paper. Discard the precipitate on the filter paper. We are only interested in the liquid which we are getting in the beaker below. The liquid is containing dissolved phospholipids. Now, once all the liquid is filtered, it's poured into a petri plate and the solvent is allowed to evaporate to dry completely at room temperature. So after evaporation, it's the crude lecithin. Next is the purification of crude lecithin. For this, add enough quantity of petroleum ether that is completely able to dissolve the crude lecithin. So lecithin is dissolved now. So how we are going to separate it. We are gonna add acetone into this again, because in the presence of acetone, the lecithin is precipitated. Wait for some time so that lecithin can get precipitated and settle down at bottom of Petri plate. Now we need to discard the acetone. You can notice pure lecithin. To make it more pure, we can repeat the purification step. This was all. Thanks for watching.